Now we will start with the Unit 9, that is Molecular Basis of Inheritance. In this unit we will study about the genetic material that is responsible for the inheritance process, which processes are associated for the inheritance. How does the inheritance in human beings occur? So let us first of all begin that what forms the base of the inheritance. As said earlier, basically genetic material forms the base of all inheritance processes. So here we will study DNA that acts as genetic material. In the previous sections we have already studied about DNA, that is what is DNA and how it can be said as a genetic material. The genetic material of any cell basically consists of various parts like DNA, RNA, constituting chromosomes, nucleus and thereby forming cells. Here we will study the DNA which acts as the main genetic material for inheritance. As said earlier, the chromosomes mainly consist of or are made up of nucleoproteins. Which means nucleo plus protein, that is nucleic acid and protein. Thus, it is because of these two that the mechanism of inheritance and thereby heredity information occurs. This nucleic acid involves the genetic material or contains the genetic material, that is DNA, RNA. Out of this, DNA is our main focus to study because in all animals, or rather say mostly animals, DNA is found to be the main genetic material out of the two. Except a few like viruses and plants. Because viruses and plants contain RNA as the main genetic material and therefore the process of heredity information is carried out within viruses and plants through RNA, while in all other animals DNA acts as the main genetic material which is responsible for the process of inheritance to be carried out. Now after studying about what is DNA, it was necessary for the scientists to search that how does the genetic material occurs in the chromosome and where it is found. Because it is now that we have proved that DNA acts as a genetic material which is present within the chromosome, but initially in order to confirm the presence of DNA within the chromosome, various experiments were carried out. Out of which the most important that is to be understood is Griffith experiment, which was carried out in order to study the bacterial transformation. And therefore, this experiment is called bacterial transformation or Griffith effect. This experiment was first carried out by a scientist named Griffith in the year 1920, where he carried out a series of experiments with or using bacteria known as pneumococcus bacteria. Thus, this process of bacterial transformation was studied with the help of pneumococcus bacterial strain. In this experiment, Griffith took two strains of such bacteria, namely virulent strain. Although bacteria he took was of the same kind, that is pneumococcus bacteria, but two different strains. First virulent and second avirulent strain. Virulent strain was of nature or was depicted by S3, while avirulent strain was R2. The properties of these strains was that the virulent strain bacteria possessed smooth polysaccharide capsules with smooth colonies. That is, they gave smooth colonies. On the contrary, the avirulent strain of bacteria, that is R2, possessed no capsules and gave rough colonies. Thus, this was the basic difference between the two strains. However, apart from this, these two strains differ mainly in the virulence property, that is antigenic property. It possessed antigenic property, that is virulent strain, which can cause disease pneumonia. On the contrary, the avirulent strain did not contain antigenic property. And as a result, it cannot cause any disease. Now, this experiment that was carried out by Griffith was to study transformation within the bacteria using these two different strains. This figure depicts the bacterial transformation within the mouse in which it was carried out, which is also known as Griffith effect. In order to study this bacterial transformation, the two strains of pneumococcus bacteria were inserted into the mouse in order to study the demonstrating effect of this experiment. As it can be seen from the figure, there were 4 cases. In the first case, the virulent strain, that is S3, such a bacteria was inserted into the mouse. Since it possessed antigenic property, it caused a disease, and as a result, the mouse died. The second instance in which avirulent strain, that is R2, was inserted into mouse. Since it did not possess antigenic property, therefore mouse did not die. The third effect in which the virulent strain of bacteria, that is S3, was heat killed and then it was inserted into mouse. Since it was heat killed, then it lost the antigenic property. As a result, the mouse lived. But in the fourth instance, when the avirulent strain of bacteria was heat killed, then the mouse died. Now, why did this occur? Because the avirulent strain, after heating, possessed or developed the antigenic property within, and as a result of which mouse died. Thus, in Griffith effect, 4 instances were considered. In first, the virulent strain of bacteria, that is S3, was inserted into mice. As a result, the mice died. since it possessed the antigenic property. Following, in the second effect, the avirulent strain of the bacteria, that is R2, was inserted into mice since it did not possess any antigenic property. When inserted into mice, that mice did not die, that is, it lived. Following the third instance, that is, when virulent strain S3 was heat killed, that is, by applying heat at high temperature, that strain of bacteria was killed. When inserted into mouse, it lived. And the fourth instance, when the same avirulent strain, that is R2, was inserted into mouse after heat killed effect, then that mouse died. Now, in the last two results, since these were two different strains, still the mouse lived or died depending on the condition that was totally opposite to the previous one. Why did this occur? In the previous two instances, the result was very obvious as decided. But in the second instance, when the strain was heat killed, why did the effect occur entirely opposite to the previous one? In the first one, during the heat killed effect, when the virulent strain inserted into mice, at that time, this virulent strain had no antigenic property. Because due to heat killed effect, the virulent strain within the virulent, that is S3, got destroyed. And as a result, the mice lived. Moreover, the analysis of this dead mice or lived mice showed that the virulent strain responsible for the transformation of avirulent into virulent bacteria or virulent into avirulent bacteria after heat killing effect gives rise to the transformation effect. Similarly, it was the case of avirulent strain. When it was heat killed, inserted into mice, it died. Because due to heat killing effect, the virulence property which was lacking in such an avirulent strain got reserved, and as a result it possessed antigenic property. That is, it had the capacity to cause disease to the mice, and therefore the mice died. In this manner, the transformation of two different strains occurs in an opposite manner. Such a process was referred to as bacterial transformation. Thus, in bacterial transformation, after heat killed effect, results into the transformation of two strains that is the transformation of the two strain effects opposite to the normal. Such a bacterial transformation was carried out with the help of DNA, which was carried as a genetic material, which was first analyzed by Griffith, and therefore it was termed as Griffith effect. Following it, many scientists helped in the suggestion or supported the Griffith effect in a more advanced manner. Few scientists were McCarthy, Avery, and McLeod. These were the scientists who, in 1944, supported the Griffith experiment. By various molecular explanation. In this, that is the experiment which they carried out to support Griffith effect, they found that DNA that was isolated from the heat-killed strains that is heat-killed smooth, that is S bacterial cells, when added to rough cells, that is R cells, the bacterial cell within which such insertion was made changes their surface character. Entirely opposite, that is from rough to smooth, that is from R to S, which also made them virulent. In this manner, it was suggested that the Griffith effect is completely true. Moreover, apart from this, DNA was the basic genetic material which helped in such bacterial transformation. Thus, DNA was the genetic material which was responsible for carrying out bacterial transformation. And so, according to this, it was thus proved that bacterial transformation involves transfer of DNA from the dead to living bacteria that expresses the character of dead cells. And such a transformation was named as recombination. Thus, recombination that was defined by the scientist was the bacterial transformation that involves transfer of a part of DNA from dead bacterium to the live one. Expressed the character of dead cells. In this manner, this type of bacterial transformation is known as recombination.